Hello, buddies. Hello, everyone. How are you all? Hope everyone is doing good. So, on the request of my students, so today we are discussing this booster class, and this is the biomolecules which we are discussing today. Hi, how are you? So, we are meeting so early today. Good morning, everyone. I guess it is good morning at your places, right? Good morning, everyone. How are you all doing? Hi, Kushi, Adhina, Meghna, Samikta. Acha, it is good afternoon there. No, Adina, I can't hear you. You're not audible. Is it morning there? So, how are you all? So, we are meeting in this booster class. I'm saying the booster class after very long time, and this session is most requested session by my students. Oh, me, good morning. Okay, okay. So, it is 11:02. Okay, excited, Adina. What about other students? Let them join fast, and then we will proceed. And as discussed, see this time it will be a revision session, and I have the notes with me, and I'll be explaining you everything. Now, this chapter is all about carbohydrates, protein, fats, lipids, and also the nucleic acid and enzyme. We'll be discussing everything. So you have to be with me. We'll be going fast, but I will definitely make sure that each and every topic should be 100 to 200% clear to you. Okay? Yes? Okay. So let's start. Come on, buddies, join fast. So how's your weekend going on Saturday? How's your weekend going on? Great. Okay. So let's start. So there are lots of topics which we have to complete today. That means first thing, how to analyze the chemical composition. See, each and every line of NCRT we will be discussing. Primary and secondary metabolites, carbohydrates, their classification, amino acids, proteins, enzymes, polysaccharides, structures of protein, nucleic acid, metabolism, lipids. All these topics we will be discussing one by one. So let's start. First, buddies, let's talk about biomolecules. So, what are biomolecules? What are, if you have to define biomolecules, how will you define biomolecules? So, bio means living, molecules means molecules only. And these biomolecules are those which are carbon containing molecules important for the life processes. They are the carbon containing. So, they are having carbon containing. So they are important for the life mechanism. Proteins, carbohydrate, lipids are there. Here, in earlier definition, that means the first definition which was given related to the biomolecule is the carbon containing biomolecule, which is having, earlier the definition was CHO. And the formula, let's not talk about it, right? So these are the carbon containing biomolecules. And other than carbon, hydrogen and oxygen is there. So they are the carbon containing molecules. Examples are carbohydrates, proteins and lipids. Yes, in this chapter we will be talking about enzyme and nucleic acids also, because enzymes, they are the derivatives of protein majorly. Now, what is a biochemistry? Later on you will be having in MBBS, one of the subject is biochemistry. So what is a biochemistry? Biochemistry is that field of science, that is a branch of science which deals with the chemical, physiochemical reactions. Chemicals and the physiochemical reactions supporting life process, which are important for their life process. Clear? These two words, they are clear, biomolecules and biochemistry. Now moving on to the next. Now here, please remember one thing. This is a very important table. Now look at this. Now the table which is given in NCRT is something like this. A table is given over here, like in NCRT. This is from your NCRT, right? Now, hydrogen is there, carbon, oxygen. Now, what this comparison is all about? So, comparison says, when we talk about the earth crust or we talk about the human body, earth crust or human body, the chemicals, they are same. That means here also hydrogen will be present. In the human body also hydrogen is present. Here carbon is present in earth crust. Similarly, carbon is also present in the human body. Oxygen, nitrogen, sulfur, sodium, all these substances, they are present in earth crust as well as in human body. The question arises, what is the relationship between these two? One relationship is, because we are the living organism, we depend on plants for the food, and plants depends on soil for the food. So overall we can say these substances, they will be present in the soil and they will be also be present in the human body as well. This is what your NCERT talks about. I'm not saying the composition is same. I am saying they are made up of similar kind of thing. Now, one of the important NEET question which has been asked from this topic is, out of all these things, one which is present in maximum amount, what will you say? Ma'am, it is the oxygen. See, question will be asked related to human body. Question will not be asked related to the earth crust. Because we will be talking about human body. We are studying zoology. So here in oxygen, 65% of the body weight. 65%. The second we are having is the carbon. Third we are having is the nitrogen. Fourth we are having is the calcium. And the last is hydrogen. So this series can be asked, like what is present in maximum and what is present in the least amount. That's it. Otherwise, there is no need to focus, no need to remember these complex things, 65.5, 1.5, no. Just remember this series. That's it. Now, what else your NCERT talks about? Listen very carefully. In the next topic of the NCERT is the chemical analysis. When we talk about the human body or the cells or the any living cells, we talk about what different type of biomolecules are present. So that is done with the help of the chemical analysis. Let's understand everything, guys. We are going to understand. We are going to do that with the help of a chemical. And don't worry, you all will get these notes. Do not write anything. This just 1 hour. Please listen to me, and I'll make sure each and every topic will be very much it will be like very much clear to you. Now, when the chemical analysis was done, now what is chemical analysis? Chemical analysis, that means they want to know the chemical composition, that what all different type of chemicals are present. Now, living tissue. What you're going to do is, let's say Aisha is going to do this experiment. Aisha wants to know that what all substances they are present in a living organism. Now, Aisha will be having two different things. She can focus on plants, she can focus on animals. Depending on the availability, she will take up that tissue. Listen very carefully. Here in the living tissues, organic substances will also be present, inorganic substances will also be present. Organic will also be present, inorganic will also be present. Now, one of thing is, Aisha first of all wants to know about the inorganic substances. Aisha wants to know about the inorganic substances, that what all inorganic substances are present. Now, what will Aisha do? Aisha will take a leaf, and in that leaf, she wants to know what all inorganic substances are present. What will she do? First thing is, she is going to dry the leaf. She is going to dry the leaf for inorganic analysis. What will happen during the drying? Water will be removed. Water will go away. Now, we will be having a dried leaf. Then she is going to burn this. Yes. In the chemistry lab, we have a thimble by which we can, in which we can burn. That is possible. So she is going to burn this. Whenever the burning occur, carbon-containing compound, the carbon will go away in the form of carbon dioxide. It will release in the, that means carbon will go away. Majority what is present? Carbon, hydrogen and oxygen. Right? So carbon will go away in the form of carbon dioxide. Oxygen will also go away. Hydrogen will go away in the form of water. What will you get? You will get a coal. Coal means the kajal which you apply in your eyes. Something like this thing you will get. So that will be rich in inorganic substances. And later on, with the help of various processes, various tests are there, not mentioned in your syllabus, you will be able to get to know sodium, magnesium, calcium, these things, they are present. So hope Aisha learned today how she can calculate the inorganic substances. That's it. By drying. Clear? Very nice. Now, moving on to the next part, let's talk about, now we have another student, let's say Samikta. Now she wants to know about the organic substances present. Now, what all organic substances? Organic substances means buddies, proteins, the carbohydrates, the fats, the nucleic acid, the amino acids. These are the organic substances. Now, she wants to know, Samikta wants to know, how can we do that? Now, she is there in a biology lab. She will find a liver piece. Any, either a leaf, because we are zoologists, so we will be focusing on the body tissue. Suppose she found one liver. वैसे I know it is not possible to find a liver, but yes, in biology lab it is quite possible. You will get a rat. From the rat you can take out liver. I know it sounds very bad, but this is what you will be doing in your MBBS as well. So what will she do? She'll take a, in Hindi we call this dori sota. I don't know what you call it. It's a pastel mortar. So in pastel mortar, she's going to make a chutney. She's going to grind it. And when she will grind, definitely she will need some water for the grinding. So she is going to add trichloroacetic acid. What will you add? Trichloroacetic acid. Normally, whenever you grind something, you need some water. Similarly, in mixer grinder also, buddies, we need some water for the grinding. So trichloroacetic acid she added. Now, later on, what she is going to do is, she is going to filter this. This is a conical, like flash, funnel she is going to take. She will take one funnel. Funnel ke upar, here, she placed a muslin cloth, right? Filter paper, muslin cloth she put. And then here, a empty beaker was placed. Then she poured everything over here. Some things will remain over here. That means a bigger substances will remain over here. Some smaller one will come over here. So smaller one will be termed as filtrate. Bigger one will be termed as retentate. Bigger one, retentate. And when Samikta saw that what is the size of the substances found in the retentate, she said, ma'am, the size is bigger than 800 Dalton. So for organic substances, the size is measured in Dalton. So in NCERT, it is 1000 Dalton written. So you can take in data of 1000 Dalton. 1000 Dalton. Right? She found the size is bigger. And because they were insoluble in acid, what did she add? She added acid. So because they were insoluble pool, this was an acid insoluble pool. When she looked into this, when Samikta did the experiments further, she saw protein is also there in this, polysaccharides are there, nucleic acids are there, and fats are also there. She was shocked at this time. She saw even fat, they are having a size less than 800 Dalton. In fact, very less size is there. Why it is found over here? The reason is fat is always found in the form of aggregate. Like this is a fat molecule, this is a fat molecule. So all of them, they are present hugging each other. So they are present in close proximity to each other and they form a big molecule. That is the reason they will be found over here. So fat have size less than 800 Dalton but present in the form of aggregates. That is the reason it is present over there. What about the filtrate? So filtrate, she saw some chemicals are there, some organic substances are there, and she saw the size is less than 800 Dalton. Size is less than 800 Dalton. Acid soluble, because they were soluble in acid, so they came down. Size was also less. Now she found sugars are there, like monosaccharides, very small. You know carbohydrates, what's the basic unit? That's a monosaccharide. So she found monosaccharides are here, but polysaccharides are here. This is the monosaccharide, this is the polysaccharide. Here, amino acid will be present. Here, proteins. So that means smaller substances will be present over here, monomeric substances will be present over here, the polymeric you will find over here. The nucleic acid will also be present over here. Nucleic acid. Now, listen very carefully. Nucleotide will be present over here. Nucleic acid will be present over here. The nucleic acid is made up of small nucleotides joined together. Here, nucleotides are present. Clear? Now, one of the questions which has been asked from this topic in NEET is, what about cell organelles? What about cell organelles? Where are you going to find the cell organelle? Because here cells are there, jinki banai aapne chutney, right? Cells are there and you made a paste or slurry out of it. So where are you going to find the mitochondria, the ER? Where are these present? They will be present in the acid insoluble pool. So that will be found in the acid insoluble pool. Clear? Is this topic clear? Right? Cell organelle. Acid insoluble pool. Clear? So here in cytoplasm, some will be acid soluble pool, some will be acid insoluble pool. Now, next topic is, when we talk about when Aisha was doing the experiment of inorganic substances, she saw sodium ion is there, potassium ion is there, calcium, magnesium, water, NaCl, such things are there. Question will not be asked from this topic. Congratulations, experiment successful. Yes. Now, in your NCERT, another important topic is primary and secondary metabolites. So what are metabolites? Metabolites are those substances which are present in the cell, right? They are basically organic substances, or organic substances. They are termed as, another name of organic substances is metabolites. Now, there are some metabolites which are super important for the body, without which you cannot live. You cannot live. This is important for your growth, for the survival, found in majority of living organisms. What are these? Carbohydrates, proteins, lipids, etc. Primary. They are important for your survival, buddies. They are primary. Now, what about the secondary? Secondary means they are present in the body, but they are not important for the survival. Are you getting this point? That means they are providing some extra properties. If it is present, that is well and good. If it is not present, that is also fine. It doesn't mean I'll die. So they are the only thing which they have seen. Actually, your NCERT says their you can say importance is still unknown for secondary metabolite. Now, they are found in microbes, fungi and plant. So there is a list from which is all-time examiner's favorite. Again, I'm writing, this is all-time examiner's favorite, and question from this topic has been asked like, on one side you will be getting some pigment, on another side you will be getting their names. So you have to match it. First is pigments. Some pigments are there which are, if they are present, that is good. If they are not present, that is also fine. Carotenoids and anthocyanin. Alkaloid. Second is alkaloid. In alkaloid, we have an example of a morphine and codeine. So, in earlier time, when at the time of war, fight. So, at that time, people, the soldier, they used to get injured, and while injury, when they are injured, so they used to feel pain. So, from the plants, these alkaloids are extracted, such as morphine. Morphine, even today, morphine is used as painkiller. So at that time, people used to take morphine as such. But today, in the painkiller, we have morphine, painkiller. Next is the codeine. Codeine is, we are using in cough syrups. Codeine is present in cough syrup. So it gives a sedative effect also. Means neend aana types effect. You remember on, if you have a cough syrup, so cough syrup ke back mein you will find a statement is written, do not drive after taking cough syrup. So next is terpenoids. Some terpenoids are there. So T for terpenoids. So monoterpenes and diterpene. T for terpenoid, monoterpenes. Here also T, diterpenes. Here also T. This is how you can manage. Essential oil. We all love essential oil massage these days. So in fact, this is important. Nowadays, we know they are important for our body. Ayurveda is based on essential oils. Lemongrass. But why it is present in plants? Still, that is unknown. Toxins such as heparin and ricin. Lactins are there. Lactins are the blood cholesterol lowering agents which we are using nowadays. That is concavalin A. Next is some drugs are also isolated. Why they are present in plants? Still unknown. Vinblastine. Vinblastine is used for the treatment of cancer. Curcumin is present in turmeric. So these are some drugs. Some polymeric substances, rubber, gum and cellulose. Cellulose is a polysaccharide. I'll be discussing that. So all these are the secondary metabolites. Plant function, very important. Again, I'm saying, please try to write these things once or twice. You will not at all forget. Okay, now Moving on to the first topic, which is the carbohydrate. So slowly and slowly we'll be proceeding towards all the biomolecules. Carbohydrates, name itself indicate hydrate of carbon, but this was the old definition. Now here in this definition, the formula which was given by the scientist was CnH2nOn. This was the formula given. Now here please remember, all glucose doesn't show the same formula. All the carbohydrates, all the monosaccharides, they do not show the same formula. For example, deoxyribose, I'll be showing you the structure. So they do not follow this formula. So it doesn't mean they are not monosaccharides, but they are monosaccharides. So this was the old definition. So later on, scientists, they gave another definition. Now the scientist which they gave definition was polyhydroxyaldehyde or polyhydroxy ketone. So the second definition was given. The most important definition is polyhydroxyaldehyde or polyhydroxy ketone. That means polyhydroxy means many OH group they have. What do they have is many OH group. And aldehyde, that means aldehyde group is present. Polyhydroxy ketone means many hydroxyl group, OH group is there, or ketone group is present. In this, the simplest formula of the carbohydrates, I am saying simplest formula, is glyceraldehyde. So the minimum is 3 carbon. Minimum they should have 3 carbon. So 3 carbon, 1, 2, 3. Polyhydroxy, 2 pe will be having OH group. Third pe we need aldehyde. Answer is glyceraldehyde. The simplest carbohydrate. Please remember, for showing the property of the carbohydrate, we should have a simple carbohydrate in 3 carbon structure. 3 carbon. No pe we are having OH group. On the third one we are having the aldehyde group. Glyceraldehyde. Happy everyone? Glyceraldehyde. Now similarly, polyhydroxy ketone, the simplest one. Here also, see, ketone will be, you know chemistry better than me. So ketone will be present in the center, C double bond O. On one side OH, here also OH, and this is. So the name is dihydroxyacetone. And formula to bhai, you are very intelligent. I guess I don't need you guys to. I don't think that I should explain you this. Please remember, number of carbon. Please remember, very important, number of carbon. Single unit, I am saying single unit, minimum 3 carbon should be present. Minimum 3 carbon should be present. Maximum 7 carbon. After 7 carbon, they will not show the property of carbohydrates. Clear? Now, if 3 carbon are present, trioses. Look, come to this. So these two, kahan gaya? So dihydroxyacetone, ये रहा. Glyceraldehyde and dihydroxyacetone, they are the example of trioses. 3 carbons are there. Triosis. Now, triosis. If 4 carbon, erythrosis. 5 carbon, pentosis. 6 carbon, hexosis. 7 carbon, heptosis. That means in one structure, minimum 3 carbon, maximum 7 carbon should be present in one structure. I am saying in one monomeric unit. Now another important property is chiral carbon. Chiral carbon is the one where different groups are attached to the carbon. Now listen very carefully. This is a carbon. Listen. This is a structure of glyceraldehyde. This is a structure of glyceraldehyde. Structure of glyceraldehyde. Now here buddies, look at this. This is a chiral carbon. One aldehyde group, one alcoholic group, one H, and this is CH2OH. All different chiral carbon are present. So that is the reason they are optically active, right? They can rotate the plane of polarized light. When chiral carbon is present, so they can rotate. Next is, most of the sugars, they are dextrorotatory. Dextrorotatory because they are having chiral carbon. So they can rotate the plane of polarized light towards the right. So that is a dextrorotatory. Except fructose, which is levorotatory. This is important question from for your NEET point of view. Clear? Levorotatory. Now dihydroxyacetone, can you find a chiral carbon? No. No chiral carbon. So this is the only one which doesn't have a chiral carbon. If it doesn't have a chiral carbon, that means it is optically inactive. Okay, so let's talk about their classification. There are various kinds of classification on the basis of the functional group. On the basis of the functional group. Now, if it is aldoses or ketosis, aldoses means polyhydroxyaldehyde, ketosis means polyhydroxyketone. Now, aldoses, if 3 carbon is present, glyceraldehyde. If 4 carbon, erythrose. If 5 carbon, ribose. If 6 carbon, glucose and galactose. Ketosis, if 3 carbon, name is dihydroxyacetone. 4 carbon, erythrose. 5 carbon, ribulose. Here it is ribose, here it is ribulose. 6 carbon, fructose. 7 carbon, pseudohaptulose. Now, question from this topic, the PYQ, one of the important question. Like fructose is what? Fructose is ketose ketose carbohydrate, or it is a aldehyde? So correct answer, it is a ketosis. It's not a aldose. But is right? Glucose and galactose, they are having aldehyde group. So look at this. Galactose and glucose. Any doubt? Tell me. If you have doubt, we can stop here and you can ask me doubts. Done? Clear? Okay, great. Now, classification on the basis of product on hydrolysis. So, simplest one is termed as monosaccharides. Monosaccharides. Monosaccharides are the monomeric unit. That means isko further we cannot break. So, not further hydrolyzed. Not further hydrolyzed. So, for example, a glucose structure in the open form, this is the structure. Right now, we don't have time, so I'm not explaining you the, you can say the structure. 6 carbon are there, aldehyde group is present, polyhydroxy condition is there. Now, this can also be formed in ring form. This is the ring form. This is a pyrene ring. So, pyrene ring may, what we have is 5 carbon, 1, 2, 3, 4, 5. 5 carbon will be present in the ring and 1 oxygen will be present. 1 carbon will be present outside. This is termed as a pyrene ring. Okay. Here various examples are there. Now let's understand the examples. Now this is here, buddies, look at this. This is an ring form. This is a pyrene form of glucose, of the glucose. If OH is present above first carbon, anomeric carbon, I'll tell you why it is termed as anomeric carbon. Now, if OH is present above, this is termed as beta D glucose. Beta D. First carbon, if OH is present above. If OH is present niche towards the lower side, it will be termed as the alpha D glucose. Right? Alpha D glucose. This is termed as alpha D glucose. Otherwise, it is beta D glucose. Is this structure clear? Beta D and alpha D. Now, this is the first one. Now, just after O, just after this group, the first one is termed as an anomeric carbon. Anomeric carbon. Now, these two, they are the anomers of each other because the change is with respect to the anomeric carbon. So, these are the anomers. When, look at this, let's take a structure of a galactose. Here the structure is very much different. Now here the difference in both the structure, if you look at the structure, the difference is with respect to the fourth carbon. Here look at the fourth carbon, here look at the fourth carbon. So here the changes are not with respect to the anomeric carbon. So this is termed as a epimers. Change other than the anomeric carbon. So they are termed as epimer. That means glucose is the epimer of galactose. Here also beta. Why beta? Because OH is present above on first carbon. And when you will make a anomer, anomer means this anomer to that anomer. That means alpha D galactose. The first carbon will be opposite. Now listen, if first carbon, if changes with respect to the anomeric carbon or carbonyl carbon, they are termed as anomer. If changes with respect to any other carbon, they will be termed as epimers. So glucose and galactose, they are epimers, whereas alpha D glucose and beta D glucose, they are anomers. Anomers, please remember. All these diagrams are there in your NCERT as well. Okay. Let's move on to the next structure. That is a fructose structure. So fructose is what? This is your ketone. They are having a ketone. There's a ketose group is present. Ketone group, not aldehyde. The ketone is present. Here look at the structure. Here the furan ring is present. Though, listen, here also 6 carbon are present. 6 carbon is there. Fructose, the structure is 6 carbon. But here, when you make a ring structure exists in furan form. So furan form means 4 carbon. 1, 2, 3, 4. 4 carbon are present in the chain, whereas 2 carbon are present outside and 1 oxygen is there. Whereas in pyron, 5 carbon are present in the chain and 1 oxygen. So this is the alpha fructose. Beta fructose diagram, everybody draw in your notebook right now. Beta. Everybody please draw beta fructose in your notebook. Can you draw beta fructose in your notebook? Alpha I have already given. So the beta, the change will be with respect to the anomeric carbon. So here in from this chapter, 12th standard May, you will be having one chapter of biomolecules. So I am teaching you everything. Your 11th class biology and 12th class chemistry. 12th class chemistry, you have this chapter of biomolecules. Is this done? Is the structure done? Kushi? Meghna? Alam? Done? Great, great. So change will be with respect to the first one. Opposite. Isko make it opposite. That's it. Now, moving on to the next. Another concept which you have to understand, listen to this very carefully. If this carbon, the first carbon, anomeric carbon is free, if anomeric carbon is free, if carbonyl carbon is free, then in the lab you can do Benedict or Fehling test. If Benedict and Fehling test comes positive, that means it is a reducing sugar. It is a reducing sugar. Anomeric carbon is free. Anomeric carbon is free. It will give a positive test. That means it's a reducing sugar. It is free. It is not involved in bond formation. Now come to this point. Come to this point. Here, let's take an example of a maltose. Here, I am taking one example of the maltose. So maltose means ma. Maltose, how will you remember? It's a ma. Or in Hindi we say, I don't know whether you know or not, and like mother, when we talk about a mother, ma. कभी अपने बच्चों में difference नहीं करती. For mother, each and every child is important. So this is how you can remember. Okay, this is my way of remembering things. So here, both the structure will be same. Both the structure are same. That means 2 glucose molecules are there. So ma, 2 glucose, same structures are present. So 2 glucose, this is 1 glucose, this is another glucose. Bond will be formed as a glycosidic bond. Here, listen very carefully. Here the carbonyl carbon is involved in bond formation. Here the carbonyl carbon is free. So your this end will give, this one will give a positive test, which is that is the reason maltose is termed as a reducing sugar. And just imagine the situation. Come on, Meghna Mishra, remember the situation. Agar, if I would have made this bond in an opposite direction. Hope I am audible. Just a second. If I would have made this thing in opposite direction, just imagine opposite direction mein, then the first carbon yahan pe hota, it would have made a bond over here. Here also first carbon is involved. Here also first carbon is involved. Will this be a reducing sugar? This will not be a reducing. Clear? Is this point clear? Maltoses. Now, let's move on to the next structure. Now, here, let's come back to the structure, which is a polysaccharides. Now, we are discussing polysaccharides. So, there are various type of polysaccharides. Polysaccharides means various monosaccharides units are there. Various monosaccharides units are there. Now, these monosaccharide unit, they will be joined by, come on everyone, get ready in the comment section and give me answer. Monosaccharide units, they will be joined by dash bond. They will be joined by yes, tell me. Adina. Glycosidic bond. Very nice. Glycosidic bond. This bond will be glycosidic bond. Reducing means to reduce the Benedict and Fehling test. Non-reducing means they are not able to reduce the Benedict and Fehling test. Now, importance. Look at the importance. This can be, polysaccharide can be important for the structure. They can be important for the storage. Chitin and cellulose are the structural polysaccharide, whereas the starch and glycogen, they are the storage polysaccharide. They help in the storage. Now, this is the table, and this table you have to understand by yourself. This is a homework which I am giving you, otherwise you will not be able to complete the chapter. So, please try to understand. So, that means here the source is given, cellulose, etc. and etc. All these things are given. I'll be just explaining you the starch because this is important. You know, starch is a storage polysaccharides of plant. It is made up of two things, amylose and amylopectin. Two things are there, amylose and amylopectin. Amylose is made up of alpha glucose, where the first carbon of one glucose is attached to the fourth carbon. Now, it will form a linear structure like this. Amylopectin, on the other hand, will be forming a linear structure by 1,4 linkage, 1 of the carbon number 1 to 4 of the carbon number 2. So, it is carbon 1,4 linkage is there. Now, here another branching will also be present. So, branching is alpha 1,6 linkage. So, at branching, that means this is the first carbon is attached to the sixth carbon. So, branching is also present. Now, one question which has been asked from this topic is, now these amylose, they form a helix-like structure. Now, amylose is the only structure which forms a helix, alpha helix-like structure. Now, because it forms a spiral alpha helix-like structure, so they can hold iodine, so it can give a positive iodine test. So, this is the one which can give the positive iodine test. Rest all cannot give the positive iodine test. Now, this structure you're going to do by yourself. Okay, buddies? Clear? Now, moving on to the chitin and inulin. Chitin and inulin, these two are another type of polysaccharide mentioned in your NCERT. So, chitin, you remember the arthropod? In arthropod, exoskeleton, beetle, insect, your arachnid, your shrimp. So, their exoskeleton is made up of chitin. So, chitin is what? It's a structural polysaccharide. Here, chitin is present in arthropod. So, name will be related to animal only. That means monosaccharide unit will be related to animal only. So, in Hindi, I don't know. In Hindi, we call snake as Nag. Pure Hindi name is Nag. So, Nag is the monomeric unit. This is how you can remember. Tarika, these are the way by which I remember. So, I guess this is going to benefit you also. N-acetyl glucosamine, they are present. So, monomeric unit N-acetyl, N at the N carbon, it is acetylated. Glucosamine structure, one glucosamine, second glucosamine, third N-acetyl glucosamine, like this they are attached. So, here the monosaccharide unit is this. Second is, this has been asked, another question. What is inulin? So, inulin is a storage polysaccharide of asparagus and dahlia. Here the monosaccharide unit is fructose. Clear? Now, moving on to the next topic, that is the amino acid. Now, you will get these notes. Please go through them, and wheresoever you get stuck here, wheresoever you are having a problem, guys, I am today, throughout the day, I'll be available on Telegram. You can write a message. You can record something, whatever you want to do. I am there and I'll help you. So, this is going to be your today's homework, that you will be going through the chapter once again with the help of these notes. They will be beneficial to you. Now, amino acid you will be found in acid soluble pool. Now, size is less than 1000 Dalton. Modified methane, amino acids, they are modified methane, NCERT says. This is a structure of methane. Now, on one end, if I add a R group, on another side, I add a acid. See, acid. Now, on another end, if I add a amino group, NH3, this will be termed as amino acid. Amino and acid. Now, this R group varies from one to another. This R group varies from one amino acid to another. Now, look at this. This R group can be H in glycine. This R group can be CH3 in alanine. This can be CH2OH in serine. Serine. These are the different amino acids. Okay. Okay. Amino acids, they are of various categories. On the basis of acidity, basicity, they can be acidic. Acidic means not only a single COH group. If they are having extra COH group, so that means they will become acidic. One acid is fine. If they are having more than one acid, that is, they will make it more acidic. For example, aspartic acid and glutamic acid. Next is neutral. Neutral means one amino group and one carboxylic group. So, glycine and alanine. Now, basic. Basic means they are having extra amino. Basic, one amino group is extra. Basic. So, lysine and arginine, La. Right? La. Clear? Now, there are some which contains sulfur. Cysteine, cysteine and methionine. They contain sulfur. Now, aromatic. Aromatic means they will be having an aromatic ring present. Yes, my chemistry people. Aromatic ring. Tyrosine, tryptophan, phenylalanine. If they are having alcohol, extra group containing alcohol, so they are serine, threonine and tyrosine. Now, this is super important. Now, question will be like, out of all these 4 options, select the one which is having the sulfur containing. Okay. Very important property is that they show Zwitter ion. Zwitter ion means they are dipolar in nature. Dipolar in, see, this is dibasic. This also exists in this form. This can also exist in this form. This is a Zwitter ion. So there is a particular pH at which both of them, they are present in ionic stage, cationic and anionic stage. So this is the Zwitter ion. Now here one is present in cationic stage. Here one is present in anionic stage, anion and cation. So, but they can, at a particular pH, they can exist as Zwitter ion, bipolar. 2 different ions are present. Okay. Now let's talk about proteins. So what are proteins? Are the heteropolymer of protein. Now similar amino acid, they do not join together. Different amino acids, they join together. Right? The bond which is present in between the amino acid is a peptide bond. Which bond? Peptide bond. They are found in acid insoluble pool because they are bigger in size. Size greater than 1000 Dalton. Now whenever 2 amino acids join together, water molecule comes out. The bond which is formed is termed as a peptide bond. It's a peptide bond. Clear? Now one side of an amino acid, N terminal will be free, because its C terminal is attached over here in the bond formation, peptide bond. So CONH is a peptide bond. On another amino acid, COH will be free. So that will be termed as a C terminal. That is a C terminal. Okay. Now, these amino acids, they are present in various structures. If it is present in a linear form, that means one amino acid joined to another amino acid, joined to another amino acid, like this they are attached. So it's a linear amino acid. It's a linear chain, right? Here the number of peptide bond, total number of amino acids, say if we'll -1, we'll get number of amino acid. Number of peptide bond. So second is the secondary structure. Secondary structure can be found in alpha helix. Now primary will fold itself into this helical structure. Second is they are present like this, plate like structure, alpha helix or beta plated. Now imagine the situation. Let's make the structure more complex. Now if one polypeptide, I have a linear chain of polypeptide. Look at this, I have this wire. Now this is a linear chain. If this linear chain is present like this, secondary structure, if I'll make a bunch of this 3D structure, a 3D, something 3D I'm making, some bunch like structure. This is a 3D structure. This is a 3D structure. Now just imagine this. If I have these two chains, and if I'm joining these two chains together, this will be termed as a quaternary structure. This will be a quaternary structure. When I am saying just one chain, a 3D structure is present. This is a 3D structure. This is a enzyme. This is a enzyme. So it forms a 3D structure. 3D structure. So this is a 3D structure. Here, myoglobin is a very good example of the tertiary structure. Myoglobin. Myoglobin. And here it is the hemoglobin. 4 different, like this is a 1, 4 chains are present, right? Hemoglobin, 4 different chains are present. Various bonds will be there which will keep the structure at their position. Now, moving on to the next topic, that what is enzyme? What are enzymes? Let's understand this. So enzymes are what? Enzymes are the biocatalyst, right? Biocatalyst which increase the rate of reaction. Now, it increases the rate of reaction without being consumed in the reaction. For product, when it reacts with enzyme, it will give, opposite. Substrate when react with the enzyme, it will give a product plus enzyme again. Clear? Now, enzyme require a particular temperature, pH, various conditions they are required. Mostly enzymes, they are made up of proteins. Protein, that too 3D structure. Very important question. So when a protein chain will fold itself, it will form a site like this, which is termed as active site. Which is termed as active site. So active site is that site where the substrate joins. So this is the active site. Look at this, substrate binds. Another site is also found, which is the allosteric site. Okay. Now in NCERT, one comparison has been given. What is enzyme? What is inorganic catalyst? Both of them, they are important for the reactions happening in the body. Now, enzymes are always living. Mostly they are made up of protein. Inorganic catalysts, they are non-living and they are non-proteinaceous and they are metal ions. They are specific in function. That means one enzyme is required for one reaction. Non-specific, they are non-specific. Inorganic catalysts, they are also used. For example, palladium. At high temperature, because enzymes are made up of protein, so protein will lose the chain structure, so denaturation, except those enzymes which are thermophilic. That means they are stable at high temperature. In 12th standard, we will be talking about one Taq polymerase, which is an enzyme isolated from hot spring, bacteria, Thermus aquaticus. In 12th standard, we have those things. Now, here, either you increase the temperature, it will not affect the catalytic activity. Okay. Now, how does an enzyme function? Let's have a look. This is an enzyme having the active site. Substrate will come and bind on the active site. There will be a transition stage. Then the enzyme will come out. The product will be formed. One example has been given in NCERT, like when carbon dioxide joins with a water molecule, it forms the H2CO3. Almost 200 molecules are formed per hour. This is the reaction rate. But when the same reaction occur in the presence of carbonic anhydrase, you remember this we have done in the chapter breathing and exchange of gases, same enzyme. So when it is done, 6 lakh molecules are formed per hour. Look at the difference. Clear? Now this is going to break up into H plus and HCO3 negative. Here also the same enzyme is used, which is carbonic anhydrase. Rate of reaction, if you have to calculate the rate of reaction, how will you calculate? How much product is formed per unit time. How much product is formed per unit time. Rate of reaction. Now tell me, any doubt? So enzymes, proteins, sometimes yes, they are made up of RNA. For example, ribonucleases, they increase the rate of reaction. Without enzyme, and the reaction is slow. With enzyme, reaction is fast. They are having an active site and they are having an allosteric site. Now, very important graph, activation energy. Now, how will you remember this? Each and every substrate, they require some potential energy to form a product. Now, if I tell Meghna that this is a mountain which she has to cross, Meghna will say that I need a energy drink. Meghna will need an energy drink. I'll give one energy drink to Meghna. Meghna will go, or she'll come over here. Then she'll say, Ma'am, I need more energy. I'll give another energy drink. And then she will cross and she will come back here. Clear? So this is the energy which is required. Potential energy is required. Now, if I tell Meghna, you have to cross this, but I'll reduce the activation energy. I'll reduce the size of the mountain. Now, Meghna is happy. That means within one bottle, within the one can of energy drink, she will be able to cross the barrier. This is the activation energy barrier. So when the catalyst is present, the activation energy is high. That means the mountain size is high for Meghna. When I'm saying I have used one enzyme, now the mountain size will come down. This is termed as activation energy. The amount of energy required, the amount of the 2 bottle of energy drink required by Meghna to cross that mountain, this is termed as activation energy for Meghna. Otherwise, this is amount of energy required by substrate to convert into product. This is termed as activation energy. And enzyme reduces this activation. Clear? Now look at the situation. From this reaction, you have to tell me whether it is exothermic or endothermic. Another point. Here, look at this. Substrate is present at high energy level. Product is present at low energy level. So, this is exothermic. Energy will be released. Delta H will be released. Now, if the product is present over here, this would have been an endothermic reaction. So, this is a chart for the activation energy. Any doubts? Meghna? Happy? Clear? Yes, Meghna said, I am clear. Meghna said, ki, ma'am, I'll be able to cross the barrier without without any energy drink. Yes? Now, there are various factors which affect the enzymatic activity. So, for an enzyme, a particular pH is required. If pH is specific, the enzyme activity is highest. At a particular temperature, enzyme activity is highest. Now, very important thing is this graph, substrate. If I am saying I am having 1, 2, 3, 4, 5, 6 enzyme. How many? 1, 2, 3, 4, 5, 6. If I am saying 6 enzymes are there. And if I am saying I am increasing the substrate, एक substrate डाला, I have added 1 substrate, enzyme activity will increase. 2 substrate, 3 substrate, 4 substrate, 5 and 6. Now, enzyme activity, that means the reaction rate will increase. More substrate, rate of reaction will increase. Now, just imagine this situation. Now, all of them, they are attached to each other. Now, look at the 7th substrate. If 7th substrate, if you will increase the further substrate concentration, what will happen? Nothing. Because all the enzymes, they are involved in the reaction. So, further increase in the substrate concentration will not affect the velocity of reaction. Clear? If you are adding 1 lakh molecules, if 1 lakh molecules of enzyme you have added, 1 lakh substrate you have added, slowly you are increasing the substrate and you have reached up to the 1 lakh molecule, the rate of reaction will increase. But the moment you will cross the 1 lakh molecule, the rate of reaction will become stable. It will become stable. They'll wait for the enzyme to get free, then they will be attaching itself to the substrate. So, this is the maximum Vmax, maximum velocity Vmax. This is a Vmax. Now, there was one scientist. So, whenever we are having a scientist, we are having some, they give us some numericals also. So, biology maybe, they give us lots of experiments, lots of questions to solve. But in your syllabus, the questions or the numericals are not there. So, one scientist came. He said, okay, you told us about Vmax. I'll tell you one more word. Now, what is that? That is a Km. So he came over here. The scientist name was Macleay's Menten. So he came and he said, I am discovering a new thing, which is Km. Now everybody was like, what is Km? So Km is that substrate concentration at which Vmax is half. That's it. At which the Vmax is half. This is the Km. So this was introduced, substrate concentration at which the velocity of reaction is Vmax. Km is a substrate concentration. Now why this came into existence? The reason is which numericals we were supposed to solve. Very important. So guys, there are two topics, lipid and nucleic acid. These two topics I have already explained you guys in the previous booster class, so that I'm not taking right now because we don't have time. I have those notes also. You will get those notes in the form of PDF. Kahin pe, at any point of time, raat ke 12 baje, at any point of time, you are stuck somewhere, you are going to write a message to me. I am available. Clear? Otherwise, I know your exam is there, the midterm examination. So, wahan pe, at any point of time, you are stuck. Please let me know. Okay? Okay? So very important, jo bhi important, whatever the important topics are, I have explained you everything. Let's talk about the competitive inhibition and non-competition. Competition. So competition is like competition between Samyukta and Alam. If there is competition in between Samyukta and Alam, so two competitors are there, this one and this one. Now, Samyukta said, I want to come first. Now Alam said, I want to come first. There is only one seat. So there is a competition. Now here, let's understand with respect to the topic. Now here, one enzyme is there. Substrate is very much similar to inhibitor. Substrate is similar to inhibitor. Both will compete itself to bind to the active site. If enzyme will bind to inhibitor, no product. If enzyme will bind to substrate, the product formation will occur. The product formation will occur. Please remember. And suppose I'll increase the army of substrate. I increase the army of substrate. Then what will happen? What will happen? Listen very carefully. If I increase the army of substrate, the Km increases. That is velocity, that is the substrate concentration will increase. Clear? Is this point clear? Now listen again, come to this point. Two persons are there who are fighting for a common thing, right? Ek ki army we have increased. So that means there is a competition. Competition will be like, one will be having a favorable thing because their population is high. Now, how you can do that? I'll give you one example. Ek bahut very beautiful example is methanol poisoning. You must have heard in newspaper, you must have read that in newspaper many times. People in the rural areas, they whenever they take, sometimes the alcohol was not good. So normally in alcohol, the ethanol is present. Ethanol. Sometime because of the poor distillation, you know, what do we call that? Desi alcohol. So villages वगैरह में like people used to make at home. So instead of ethanol बन जाती है, right? And whenever a person consume it, it will, listen very carefully. Whenever person consume it, I have a good example. Methanol poisoning. Enzyme is alcohol dehydrogenase. Suppose the person has consumed methanol. Now all the alcohol dehydrogenase will be used. So in this case, so this is poisoning. You know, many people die because of this thing only, poor distillation. So at this time, people are given ethanol. Ethanol is an actual substrate. So when a population of this ethanol will increase, there will be competition between ethanol and methanol. So the alcohol dehydrogenase will be released. The person will be not poisoned. So this is one of the things, because ultimately if you look at the structure is almost similar. Here the two carbon are there, here the one carbon is there. Now similarly, another example of competitive inhibition is succinate dehydrogenase. This is an enzyme. The substrate is succinate, inhibitor is malonate. If you look at the structure is similar to each other. Yes? Happy? Happy? Dappy? Great. Now, next topic is, there are various kinds of enzymes. Oxidoreductase. Oxidoreductase means one substrate which is in reduced state will be oxidized, one which is in oxidized state will be reduced. Name itself indicate. Second is transferase. Transferase is I am transferring one G, one group to another. So I have one S on which G is attached. Now when I'll use the transferase, the G will be attached to the S dash. Hydrolase. Hydrolase is the one which will catalyze the group, catalyze the bond, any bond, ester, ether, peptide, and water will be released. This is hydrolase. This is hydrolase. Now, second we are having is the lyase. Lyase also help in removal of group, but here the water is not released. Here water is not released. This is the difference. Next is isomerase. Isomerase is geometrical isomers are changed. D is converted into L form. Dextrorotatory, the D is converted into L form. Isomers. Now ligase, ligase means feviquick. Two things are joined together. Ligase. So accordingly, name has been given. So they are having different names also. 6 classes may we have divided. Or further 4 to 13 subclasses are also there. Okay. So buddies, look at this. We are having the lipid topic also. Everything is here. I will provide you this PDF. If you will go through it, and whenever, because already we have done the lipid and the nucleic acid in booster class number 2, and you can watch that session at any point of time. Beta, you are stuck, do let me know. Otherwise, I have tried a lot taki I can teach you, I can tell you each and every line of NCRT. Right, Adina? So hope your problem, hope your tension is relieved. Now you have your notes. Do it right now. Okay? I want happy faces after the midterm examination. That is the reason I came up with this solution, that this is the maximum thing I can do. Or maybe I'm not promising you for that. In the next session of the booster class, or I will give you I can teach you the fats and the lipids and the nucleic acid again, if that is possible. But I cannot promise you for that. But right now, your work is left, which is going through the PDF, going through the questions that you have to do. Happy now, everyone? Major topics are done from my side. Thank you so much. Thank you so much, guys. So see you buddies in the next class. This whole PDF without removing any sheet I am providing you. Go through them. Take care. Have a good day. Enjoy Saturday and do complete the chapter. See you. Bye bye. Thank you so much.